Was just asking somebody, how did Devin get roped into this just now? Anyway, it's good to see you. Good to see you all. So I'm giving a final report, even though I just got a 1-year extension on my grant, because we're still making wine, we're still continuing, we're still collecting pruning weights. The winter has only just started, as you have noticed. So I'm just giving this final report on what we have done to date. Just briefly background. What we're doing is working on heat stress. How do plants, especially Cabernet Sauvignon in our case, respond to heat stress? And then maybe what can we do to mitigate some of the effects of heat stress? You've all noticed the last couple of years we've had some heat stress in Eastern Washington, maybe across the Northwest and down into California. So heat waves are becoming more frequent, they're getting more intense. We have shattered records last year, and then it happened again this year. So it's getting hot. Most of us who work with grapes, we deficit irrigate. We use regulated deficit irrigation of some kind to apply some kind of water deficit, because we think that's a good thing for the quality of wine grapes. But what we often don't know is how that water deficit interacts with heat stress. Does it make it worse, or do the vines even care? We just don't know enough about that. So we're going to look at that a little bit, what's the vulnerability and so on. And then finally, what are the consequences for production of grapes and the final wine quality. So we worked with Cabernet Sauvignon. First objective of this particular project was to look at acclimation potential when vines are grown in different seasons. How do they respond to a sudden heat wave? So the first objective, and then the second, a mitigation, a practical approach to try and mitigate heat stress by using a novel cooling system, evaporative cooling system that we've developed, and I'm going to present that today. And then we'll look at what the impact of that mitigation is on production and grape and wine quality. So here's the first objective. We did a couple of pot experiments. This is in climate-controlled growth chambers, where we had potted Cabernet Sauvignon that we put in these growth chambers, grew them for 10 days as an acclimation, so they get used to the growth chambers. And we simulated 2 different growing seasons, a cool season and a warm season. I'm having everything here in degrees Celsius and so on. I'm sure you can all translate because I can't. Cool season, 27 degrees during the day, 10 degrees at night, and then a warm season, 32 degrees day, 15 nights, so 5 degrees C or 9 degrees Fahrenheit higher average temperature during that acclimation period for the warm than the cool season. And then on day 11, we imposed 6 different temperature regimes. So you can see the temperature regimes in these growth chambers follows typical diurnal trends, not the classic growth chamber night constant, day constant. So we're programming these chambers to follow classical day-night diurnal trends. And then day 11, I don't know if I can use this pointer. Yep. Day 11, 6 different temperature regimes going from a little bit below what they experienced all the way to above 40 degrees Celsius or 104 degrees Fahrenheit. And then we measured gas exchange, a bunch of other things, but I'm just presenting gas exchange because I have, 15 minutes? 20 minutes. Okay. So these are what we call sensitivity curves or vulnerability curves. This is the change in photosynthesis at midday between day 10, so between the last day of the acclimation, and day 11. So this is, the vines have been happy at their respective growth temperature, and now they're suddenly experiencing a heat wave or something. And you can see that in both cases, whether they were grown under warm or cool conditions, the more heat you give them, the more photosynthesis drops. When you go a little bit below, as in this treatment here, a little bit below the previous temperature, photosynthesis actually increases up to 150% of what it was before. So this is only 100% is what they experienced the previous day, and then this is the line of how much percentage decrease in photosynthesis they're experiencing. So their gas exchange, their sugar production in the leaf drops as the temperature increases. And it's the same regardless of whether they had grown under cooler or warmer conditions. That was a bit of a surprise for us. We thought maybe with different slopes in these lines, but they're totally parallel. So same sensitivity of vines, but if they had grown under warmer conditions, a 5-degree warmer season previously, then the same photosynthesis was reached at 5 degrees higher temperatures during the heat wave. So they were able to compensate, to accommodate that heat wave better if they had already experienced warm conditions before. And that's important for us to know if we want to mitigate heat stress. It depends on what the vines have experienced prior to a heat wave. So that's an important piece of information, because we can then maybe use threshold temperatures at which we can activate, for example, an evaporative cooling system or a shade cloth or something. And that activation temperature depends on what the season temperature has been. So I'm going straight into the second objective. This is what we developed. This is our mist type evaporative cooling system. Ben-Min Chang says hi. He recently got Pat Bowen's job in British Columbia. He was the student and then a postdoc who developed this system. I call him the gadget kid because he had all these gadgets that he played with. So he had an evaporative cooling system that incorporates, if I can ever get this, can you make this bigger again, please? All right, I'll click. Sorry, wrong button. So, why is it doing that? Oh, now it's just making it difficult. Okay, will it work? 3 times the charm. So we have these misting nozzles that we installed either on a foliage wire or catch wire on the canopy, just on the west side of the canopy, because that's the warm side of the canopy usually. Or then this last 2 years, we put them on the irrigation wire below the canopy facing up and cooling either the canopy side. You can see how these misting nozzles here in the picture spray out and have this fog or film of water on the outside of the canopy that then evaporates and cools the canopy, or it sprays it up into the fruit. So you can either cool that canopy side or cool the fruiting zone. Then we have infrared thermometer that measures across the row, measures the canopy temperature, and then we have these threshold temperatures where we activate the misting. In this case, we say, okay, at 35 degrees C, 95 degrees Fahrenheit, the system comes on. Canopy temperature reaches 95 degrees, and then at 30 degrees C, or about 90 Fahrenheit or something like that, 89, it turns off again. So it only runs or mists when the temperature of the canopy is between these threshold temperatures. And it turns off again. It also turns off, we have a leaf wetness sensor here. When that sensor picks up that the leaves are getting moist, the system also turns off, because we don't want to favor diseases in the system. We want water not on the leaves, and we don't want it on the vineyard floor, because we don't want to interfere with our deficit irrigation strategy. We want the water just in the air, evaporating and cooling the canopy as it does. So that's the goal. Now, some result, and you can look at this video while I'm talking, if you get bored with what I'm saying. Here we have the ambient canopy temperature in blue over that late July heat wave this year, 2022. You can see how hot it got, up to 47 degrees Celsius, really warm. And the misting system cooled that canopy down by 5 to 10 degrees C below that peak of temperature that we had. So the misting system worked really well. You can see it here, how it mists. There's a little bit of water that drips on the vineyard floor. We still haven't figured out how to get completely rid of that, but really the water just evaporates through that canopy and cools the canopy down. So a very effective cooling mechanism for the canopy. Water use about 1 millimeter per day, very minimal compared with the typical classic overhead sprinkler system. They use more than 10 times more water in those systems, and those are also very expensive to do. System is fully automated with solenoids that turn on and off the water supply, depending on those input sensors that measure canopy temperature and leaf wetness. So this is the system in action, and then you probably want to see some results. Here is a list of things that we measured. This is not a complete list, just something that I can fit on the table. Mainly, what we found, growers are often worried that we're going to induce more shoot vigor, we're going to induce big berries with lots of methoxypyrazines if we use more water in the canopy, and that didn't happen. What did happen is that the berries did increase in size, but remember, this was in 2021 when everybody had tiny little berries. So we thought this was actually beneficial in the year where this happened, because we got 13% bigger berries in 21. We also got, strangely enough, I don't know why, we got more seeds and bigger seeds, so potentially more seed tannins in those berries. We did measure tannin in the wine, I'll show you that later. We got higher TA, just like we had expected. When you cool the canopy down, you hopefully get some higher acid, so we're preserving acidity, which is what we had tried to do. So that's for 2021. And then in 22, this year, same effect on berry size. Again, we had a very hot growing season. Our berries got quite a bit bigger. We also had more than 20% more clusters this year than last year. Actually, we had 20% more clusters in the cool treatment than in the heated treatment. So we think maybe that is a carryover effect from the cooling from last year. Maybe the cooling increased bud fruitfulness, so we had more clusters per vine this year than we thought we would. And you can see that here, cluster number quite a bit higher in the cool treatment than in the heated treatment. So overall, quite a bit higher yield because of the combination of more clusters and bigger berry size. So quite a bit higher crop yield, again in a very hot year. But no dilution effect. We had higher acidity, higher malic acid, and higher yeast assimilable nitrogen. Lots higher, up to almost 50% more yeast assimilable nitrogen in the berries that had been cooled rather than the ones that had been warm. So the dilution effect theory goes out the window. We had actually higher concentration of constituents in the bigger berries in this case. They just produced more or imported more of the stuff that they were supposed to. So the cooling increases yield, doesn't delay ripening. We found no difference in Brix levels. We harvested 25, 26 Brix, just like you would for Cabernet Sauvignon. Increased berry size, acidity, YAN, no effect on growth, no effect on diseases. I'm not showing you, we checked for botrytis, powdery mildew, physiological disorders on the clusters like berry shrivel, bunch stem necrosis, that kind of stuff. We found no difference in vigor. We haven't pruned these vines yet, so I can't tell you about pruning weight just yet. And then finally, this is wine. We only have wine composition from 2021. The wines are still fermenting this year because of a very late harvest. Overall, very, very small differences. The wines overall, when you look at these conditions or composition changes are similar, except that with cooling we had slightly lower tannins, and probably because we had lower tannin, that then translated to lower overall polymeric pigments. Anthocyanins were the same or even slightly higher with the cooling, which is what we had hoped for in this case, than with the traditional doing nothing approach. So overall, we think maybe the system has potential to work. It might help us even out vintage differences a little bit in very hot growing seasons. We'll have to see how that goes. And take-home messages. Last slide. The sensitivity to heat stress does not seem to change whether or not vines have grown under cool or not so cool, warm conditions, but they can cope with heat stress better if they have already experienced warm conditions before overall. So the slope of the change is the same, but the line overall is shifted to higher temperatures under warm temperatures. Okay. The other thing is mist evaporative cooling seems quite effective to cool the fruit zone temperature. We kind of like that idea of putting these nozzles on the irrigation wire and then spraying up into the fruit zone just to cool the fruit zone and not so much the leaves. The leaves seem to do just fine under heat for the most part. No change in vigor, disease incidence, and so on. Methoxypyrazines were the same. We essentially had none in this wine. Below the measurement threshold in many cases. Berry size was bigger, and we think maybe bud fruitfulness could also increase in a very hot year, which is when you would use the system in the first place. So higher juice acidity, higher YAN, slightly lower tannins and lower polymeric pigments in the wines. And as you remember, 2022 and especially 21 were years with extremely high tannins in some of our wines, higher than we would have wanted in many cases. So this might actually be a positive thing to be a little bit lower in a hot year. And then thanks to the team, thank you for funding this study. The Washington State Grape and Wine Research Program helped us with some vineyard management, so they also need to be thanked for funding some of this work. And then a lot of people, and this is where our water comes from for irrigation and cooling. I hope we have a lot of snow this winter. Thank you. Do we have time for a question, or are we?
Yeah. It's 1 minute for questions.
Julie.
Marcus, if you were going to scale this up, say 100 acres, what would the cost be for installation?
You had to ask. I don't know yet. I asked Ben Min to give me an estimate of the cost, and he hasn't given me that yet. He's still penciling and thinking. I mean, that is one of the things, right? How much does it cost? And I just really don't know. The other thing is, how robust is it? We don't know yet how this would hold up under like mechanical pre-pruning, mechanical harvesting and things like that. So far, we've been a bit careful to take it down before a machine goes into the vineyard. And now the next step would be to actually scale this up and put it in a commercial vineyard, which had been the plan, but that collaboration for some reason didn't work out this time. So we'll put this in a vineyard and then run machines over it and see, does it actually stay there, or does it just get shattered?
Millimeter of water used through the misters, is that on a per vine basis or an acre basis? That is just a millimeter, so it doesn't matter what the surface. So what Dai is asking is I'm sorry.
Sorry, yeah, Chad Vargas asking if they want me to repeat the question for online. Was the millimeter water usage reported per vine or over like an acre millimeter of water?
So with a millimeter, it doesn't matter. It can be any surface that you want. It's just a millimeter. So if you want to do it by acre, that's fine. It's a millimeter per acre then. And if you want a millimeter per hectare, it's a millimeter per hectare. So yeah.
Do you have any thoughts on how grapevines are acclimating to warmer temperatures? What the mechanism is for that?
Yeah, I mean, there's a lot of stuff going on. They produce, like under hot conditions, they produce chaperone proteins that help the cell membrane stabilize, so they don't become fluid as easily. Heat shock proteins, they're called. Their chlorophyll ratios change, their leaf angle changes under hot conditions, so they're not facing the sun as much, so they're not hit by as much energy. They're changing the rubisco protein, the protein that assimilates carbon dioxide. So they stabilize that proteins. There's lots of things that go on in how they cope with heat stress. Yeah. 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 So the idea would be lower respiration, therefore higher photosynthesis. That's not usually what they do. The respiration rate usually goes up more than photosynthesis as the temperature goes up. But photosynthesis acclimates, but typically the thinking is that for each degree heat that they experience, they acclimate to be able to photosynthesize at about half a degree higher. And in our case, it turned out to be 1 for 1, which is much better than we had expected.
Okay,
so now I don't know.
Have to cut it off. Marcus, thank you so much. You started a little bit late, but we went over a little bit. If you have more questions, we're taking a little break right now to grab box lunches. And so ask Marcus then. Yeah, thank you. Great job.
So just to expand on that, the entry to get into finding your box lunch is behind the registration table. It's in ballroom 5. So you guys can snag your lunches, and then those that want to participate in the berry side of the priority setting sessions. You guys can eat your lunch in ballroom 4 and then be ready for that to carry over the next few hours. Not a few hours, probably hour and a half. Sorry, I feel like I'm stressing you guys all out with that. And then ballroom 2-3 would be where the wine and wine grape side of the and table grape side of the efforts will be. So you can bring your lunches into that space. So we're splitting up for a little bit, but from the virtual side of things, that's also how it will work. So those live streams will be available 15 minutes before those sessions are about to start. All right, thank you.
It's on. Okay. Hi, everyone. Please make your way to your seats. We're going to get started. I'm going to moderate this session again. So as everyone is getting seated, this is Dr. Jana Lee from the USDA ARS in Corvallis, Oregon. And she is going to talk about her sweet SWD control and non-target effects in field trials with erythritol. I can't say it right. Anyway, I'm going to turn it over to you.
Okay, thank you for making your way over. So, as you know, spotted wing Drosophila is a problem in small fruits that's typically controlled with insecticides. However, frequent spraying can result in secondary pest outbreaks, such as with scales, and development of insecticide resistance in the flies. Erythritol is the active ingredient in Truvia. So it's sweet, but it has 0 calories. It's safe for us to eat. And a 6th grader had found out that erythritol was toxic to Drosophila melanogaster. Subsequently, spotted wing Drosophila that fed on erythritol are found to die, as well as other flies, termites, psyllids, and spider mites. Manyang paired erythritol with sucrose and found that they feed more on it. They essentially get very thirsty and lose water. While the fly is still alive, they might have eggs in their ovaries, but if they fed on erythritol, they don't lay very many eggs. So the erythritol is just causing them to hold their eggs inside. And the erythritol also acts in other ways, in that the fly will avoid laying eggs on treated fruit and prefer untreated fruit. Oh, in this example where they still hold on to their eggs, they're given normal blueberries and they still don't like to lay as many eggs. Okay. When erythritol is sprayed in the field, it can reduce spotted wing Drosophila infestation by 64 to 93% in trials in blueberries in Mississippi. This was done with 3 weekly sprays. We tested the 2 molar erythritol solution with sucrose at first, and it resulted in a 96% reduction right away, and then a 49% reduction over a 6-week period after 1 spray. So that's good, but the 2 molar erythritol solution is sticky. So Manyang worked to reduce it using a 1.5 molar erythritol. It's still toxic, it's less sticky. Now, the sucrose that's used in this formulation does provide spotted wing Drosophila with energy if they don't ingest a lethal amount. So he got the bright idea to use sucralose as a non-caloric phagostimulant. And if you see those yellow packets in the back, that's what you're eating. It's sweeter than sucrose, and we have this patented. So we had a graduate student, Bree Price, who examined sucralose. So she found that a 0.1 molar sucralose, very low dose, paired with erythritol worked well. In choice tests, the fly is given drinking straws, and they'll prefer to feed on the erythritol plus sucralose versus erythritol alone. And they also die faster with the formulation versus erythritol alone. Then she did some very tedious studies, extracting the hemolymph of the flies under a microscope. So here she had to collect it right there, and found that the sucralose accumulates in the hemolymph and they have a hard time excreting it. And so here, this happy fly fed sucrose has a plump abdomen. This poor fly fed erythritol plus sucralose drank a lot, but has a sunken abdomen. So it's losing all of its water and then eventually dying. So the objective of our project was to evaluate two of our formulations with the 1.5 erythritol paired with sucrose and also paired with sucralose when it's sprayed on fruit at the sign of first blush. And we test whether it would reduce infestation and whether these sprays would affect the fruit. And during our earlier trials, we noticed there was some leaf spotting, and so we wanted to examine if it was phytotoxic. And then there's a question whether erythritol might affect non-targets such as honeybees or parasitic wasps. And then lastly, these two formulations are very long, so the shorthand is ES or ESOL. So to test whether in a reduced infestation, there were 4 trials run in open blueberry fields, or we did one with the Elliots in the tunnel. So here we could throw a lot of flies, as much as we want, in this tunnel. And we sprayed the plots with either formulation or kept a control, and they were replicated 4 to 5 times. We collected fruit each week to measure infestation. So sadly, we didn't find any difference. Overall, there was very few spotted wing in the open field trials, and then also very few in our tunnel, despite us putting thousands of flies there. 2021 was a very hot year, and it was even hotter in the tunnel, so I guess they didn't make it. We did enclose spotted wing Drosophila, 10 females, on these sprayed or unsprayed blueberry clusters. So the females really had nothing else to do except for lay eggs or not. And they were also given fresh honey water to stay strong during the trial. And in the Rika example, we found a 39 to 69% reduction in the egg laying. And this is consistent with the past study where we use the same formulations in Elliots. To look at fruit, we counted the presence of absence of mold on hundreds of blueberries per replicate. And then we also harvested the fruit, testing them for flesh firmness on this Fruit Firm 100. And then we checked for the skin firmness, basically poking the fruit with a pressure gauge, and then looked at sweetness. So in summary, there was no difference in the fruit that was treated with erythritol or the control for any of these parameters. I have a graduate student, Abigail Greenhalgh, who's working with Dave Bryla and Carolyn Skagel to examine phytotoxic effects. So she started off in the greenhouse using a higher 2 molar erythritol in the formulations just to try to see what happens. And she sprayed potted Duke plants, and each week she scored them for visual damage. She took stomatoconductance readings, leaf chlorophyll fluorescence, which is a measure of photosynthetic potential, and then she took leaf punches to measure water content. And she also compared the cheap erythritol that we were using to a very expensive pure erythritol, just in case the cheap one had some impurities that was affecting the plants. And here you'll see there was no difference in the amount of spotting between any of these erythritol solutions, whether they're cheap or not, paired with sucrose or sucralose, but there was more spotting than the control. But if we look at the other parameters for plant health, there was no difference between these plants treated with erythritol formulations or the control. So that's a good thing. She also repeated it this summer in 3 more field trials using 1.5 molar erythritol, the usual dose. And again, there was some spotting that occurred, but there was no difference in the other 3 parameters. Third, we looked at the effects on honeybees. Previously, Ramesh helped us, and he found that adults enclosed with erythritol lived similarly as if they were given sucrose. So that's a good thing. The adults are pretty hardy. Also, Bree took measurements of bee visitation in her field trials and found that bees visited these variously sprayed bushes at the same rate. So that's good news that the bees aren't preferentially attracted to go visit the sweetly poisoned bushes more. But, you know, if you're in the erythritol sprayed plot, you will sometimes see the bees feeding on the sugar residues. And there's concern that they'll pass it to each other, which eventually get carried to the larval brood. So she did some studies where she directly exposed the bee larvae to high doses of erythritol, basically 2 microliters of the formulation or water. And she replicated this with 28 young larvae or 28 old larvae per hive and replicated in 3 hives. And then she monitored the same larvae over time. So this graph shows that the larvae survive similarly when they're fed erythritol solutions as with water. And then the main separations you see here is that the older larvae fare better than the younger larvae. And that's probably because the younger larvae don't handle the disturbance with the pipetting as well. And then here, the honeybee larvae make it to adulthood similarly when they're fed erythritol solutions or water. Our last objective examined how erythritol might affect a beneficial parasitoid. Pachycropoides vindemiae is common in Oregon and Washington, and attacks spotted wing Drosophila pupae. So here, Gracie exposed them to the erythritol formulations either directly in a vial or dip the blueberry fruit. This is the exposure rate route that we would expect in the field. And then this graph here shows in the purple line that when they're fed sucrose, that's our positive control, that they survive well. When they're just given water or the erythritol plus sucralose, they die the quickest, so they're essentially starving. But when they're given erythritol plus sucrose, they do die, but they actually, quite a few make it out for 2 weeks. So this is actually quite good because spotted wing Drosophila die much quicker with those solutions. Now, the other thing is that parasitoids prefer to feed on floral nectar or other like liquid forms. Their mouth parts are not exactly adept at scraping off dried sugary residues. So we asked what happens when there's other sugar sources available. So first, in this first scenario, they're given the erythritol formulations when the blueberry is treated. Next, they're given the treated blueberry with a cut blueberry, and in all these cases, they have water. And then next, they're given the treated blueberry with flowers, buckwheat. Other studies have shown that they benefit a lot from buckwheat flowers. And so they do live better. Many of them are alive, about 50 to 80% after 3 weeks, especially when they're given flowers. So that's a good thing. So in summary, the erythritol formulations can reduce flies from laying eggs on the fruit in enclosed situations. Then fruit that's harvested have the same flesh and skin firmness and bricks, and the leaf stomatoconductance, chlorophyll fluorescence, and water content are similar whether treated or not. And we found that honeybees visit these bushes randomly, and that larvae that are exposed to erythritol survive to adulthood. And lastly, the parasitoid wasp survives better when there's other sugar sources present, which would probably be the case. So we thank the Northwest Center for funding this and the many people in our unit who helped make the study possible, the Honeybee Lab, and our many great helpers. So thank you.
How about now? Sound good? Okay, thank you for the talk. That was really interesting. A lot of sugar substitutes have an off flavor. When you tasted the blueberries, did you notice anything different?
Well, the blueberries tasted slightly sweeter. I didn't notice an off flavor.
Cool, thank you.
Do you know how fast the erythritol degrades? Yes.
Well, it should stay present as long as it doesn't rain. I mean, do you have many more things? It's
Mhm.
or
Is there any what? No, for the fruit residue. Like any other pesticide.
Yeah,
you would
it's-
look at the-
so the main issue with the erythritol sprays is that when it rains, we have to go back and we have to go back and respray.
So Jana, in the lab trials, you saw reduced egg laying, but not in the field trials because there was just not enough pressure, correct? That's what pressure you
In the
think?
field trials, we saw reduced egg laying in the bag
Yeah,
trials, but
right,
not
okay.
the open. And in the open field trials, we had low pressure, so it was really hard to see anything.
I guess a second part to that, what rate, I mean, I don't really know what the molars, what does that translate to and what a
Oh.
grower would use in the
Okay,
field?
so it's about 26% sugar by weight for the 1.5 molar erythritol with the sucrose, and then the other one with sucralose is about 18% sugar by weight. And
Okay, thank you.
Thank you so much. Okay, Professor Laurent Deluc from Oregon State University, you're up next. Yeah.
Good to go? Great. So as mentioned, I'm going to talk a little bit about wine and wine microbiology and about using non-Saccharomyces yeast as biocontrols during pre-cold soak fermentation. So what is pre-fermentation cold soak? So pre-fermentation cold soak is where red grapes are harvested and brought into the winery, but instead of being warmed and fermented straight away, they're cooled down either through glycol jacketed tanks or layered in dry ice to a temperature typically between 5 and 10 degrees Celsius. Probably 8 to 10 is the most common. And the goal of that cooling down process is to stop fermentation from starting. So even if you don't inoculate Saccharomyces, if you're sitting up above probably 12, 13 degrees Celsius, fermentation may start to kick off naturally. So the point of cold soak is to hold the grapes at a cold enough temperature that fermentation doesn't start, and you get potentially some benefits of soaking those grapes for anywhere from a day to up to a week, or I've even heard people doing 10-day cold soak. So there's quite a lot of range in how people go about this. And there's also some different goals in why they would do that as a winemaker. One of the rationales given for this whole process is it can impact, I'm not going to say improve, but it can impact color and phenolic or tannin both composition and maybe sensory impacts. So the soaking process before alcoholic fermentation starts is thought to maybe you get some extraction of things out of the skins, out of the seeds in this water-based environment before there's alcohol around that might impact color, that might impact mouthfeel. So that's one rationale, and there's papers out there that either agree with that theory or completely disagree with it. It's very much all over the board with that, and if you ask a phenolic chemist, you'll probably get answers all over the board with that as well. The second one, I think more recent reason why this might be a beneficial thing is that the cold soak itself may impact more aromatic compounds, and that some of that is actually related to cold tolerant yeast that are present on the grapes during that cold soak period. So you're kind of providing this time period where Saccharomyces can't ferment because it's too cold, but there are other yeast on the grapes and in the vineyard, in the winery, particularly non-Saccharomyces yeast that are a little more cold tolerant than Saccharomyces that may contribute during this early phase before Saccharomyces dominates. So the changes you might see in some of the flavors and aromas of the wine may be enhanced through this process where you give these non-Saccharomyces yeast a kind of an environment to do something before you dominate with Saccharomyces and alcoholic fermentation starts. So that's kind of the two major reasons. As a microbiologist, I'm more interested in the non-sac piece of that, but definitely there still could be some phenolic color reasons why a winemaker might conduct this process. So it sounds really great, but there are some downsides to it. And one of the downsides to it is there's increased risk of some microbial spoilage occurring. So one of the reasons you might get your fermentation started quickly, you might inoculate with Saccharomyces, is you kind of want to dominate that fermentation and stop any of these spoilage yeast that might also be on the grapes or in the winery from getting a foothold. So cold soak, you're kind of opening the door a little bit to some of these other yeast. If they grow too much, or if there's too much of them in a year, where maybe there's some damage to the fruit, or you're not doing a good job with sorting, that you might actually get some spoilage issues occur during this process. In particular, there's one yeast that we worry about, and it's called Hanseníaspora uvarum, or this Clochera apiculata is another name you might hear for it. It's the same yeast. And this particular yeast is fairly cold tolerant, so it's also fairly commonly found on grapes. So if you're doing a cold soak, it is a yeast that's more than likely going to be present during this process. And its main issue is if it grows fairly well, if maybe your temperature's a little bit warm or you don't have maybe some SO2 in there to control it, it produces a lot of a compound called ethyl acetate, which smells like a very solvent, like nail polish remover. So you can get this very solventy note coming off, and if there's enough of it, it can carry through into the wine, and you can have problems from a spoilage point of view. So ethyl acetate, and to some degree, the other compound, acetic acid, vinegar, can also be problematic. This figure here from a paper really just shows, so we've got time along the axis here. 0 would be the start of alcoholic fermentation. So if we go into the negatives, that's the cold soak. And at the start right here, we have the grapes, and they've got a few different yeast on them. The black bar is Hanseníaspora. So as the cold soak proceeds, we see that black bar kind of dominate the population until you inoculate with a Saccharomyces right here, or warm up so that Saccharomyces starts growing, and then it dominates. So during this cold soak period of time, Hanseníaspora is typically the dominant yeast there. So what is a kind of standard practice for preventing this? Well, one, you cannot do cold soak, and that's often a good choice, particularly if you've got a year where there's a lot of disease pressure and maybe a higher microbial load on those grapes that are coming to your winery. But if you want to conduct cold soak still, you've got a few options. You can do a good job of sorting either in the vineyard, or increasingly a lot of sorting in the vineyard as well, looking for damaged fruit, damaged clusters that you can sort. You can use temperature to your advantage. So cold soaking at a temperature that will hopefully slow down or prevent the growth of these yeast. And then our good friend sulfur dioxide in the winemaking industry that we use for a lot of microbial suppression is also generally pretty effective against these yeasts. So we have some tools there we can use, but there is a move to, a lot of interest in moving away from some of these things, particularly the use of sulfur dioxide. And there's a downside to, I guess, from a microbial point of view, if you're adding a lot of SO2 into your cold soak to repress Hanseníaspora, you're also going to be repressing all of those other yeasts that potentially maybe are doing some beneficial things. So are there some other options there that we can look at to either maybe allow some more impact of these beneficial yeast and also, or potentially also reduce the amount of SO2 used at that point? And that's where this idea of using so much sacrificial cultures, but cultures that we would think of as bioprotectants or biocontrol cultures that we add into the cold soak to kind of suppress or control the spoilage yeast. It's not an uncommon kind of concept. It's used in other fields and other areas and other systems, just not one that we are commonly deliberately doing in wine. So that was sort of the question that we in our lab were looking at and have been looking at for a little while, is using these yeast as bioprotectants. There's a lot of commercial cultures of these particular yeasts that have got lots of different uses, and one of those uses might be a biocontrol. Some previous work in our lab, we'd shown that a lot of these commercial cultures, we tested them and saw some effectiveness at their ability to repress the growth of this yeast, Hansenia spora, as well as reduce acetic acid production and somewhat some ethyl acetate production during a Pinot Noir cold soak as well. So we had kind of a baseline of knowledge that some of these cultures, most of the cultures we tested had some effect, but we'd only really tested them under one condition that was around 8 degrees Celsius, so on the colder side of cold soak. And in our initial tests, we had not used SO2 as well, because we were just wanting to see how effective they were without this added impact of SO2. So there was still a lot of questions we wanted to kind of either optimize the use of these yeast or see how, if you were maybe in a winemaking situation where you might still be using SO2, would this still be an effective tool? So number one, we wanted to look at temperature. If we had slightly warmer temperature, maybe challenge the yeast a little bit more, would it still have some of this effective protection? And then secondly, what about SO2? So if you're going to use these cultures as a winemaker, do you just not use any SO2 at all? Or if you use SO2, are you maybe killing off the very yeast that you're adding in? So how do you maybe use these two things in combination, if at all? So the two major objectives of this work were firstly to, well, really the main objective, investigate the effect of temperature and SO2 on the ability of some select non-Saccharomyces cultures to repress Hansenia spora uvarum growth and production of acetic acid and ethyl acetate. We initially did a number of studies in like a model system where we could control a lot and narrow down where we wanted to look at. And then we took the findings from that and we did some studies with Pinot Noir grapes during cold soaks with Pinot Noir. So in the model grape juice system, our first set of experiments, we looked at different temperatures. We already had some data on 8 degrees Celsius. So we did a series of experiments at 10 degrees Celsius and 13. So 13 is really not what I would consider a cold soak, but it would be definitely a bit of a challenge for these non-sac yeast to maybe still repress Hansenia spora. So we wanted to go a little bit hotter and see how it would work. And we used we narrowed from earlier work, we kind of narrowed down to two particular cultures, Mechnikova fructicola and Turullospora delbrueckii. And so those are two commercially available cultures that we use for these experiments. And then we used the Hansenia spora uvarum that we'd previously isolated out of some cold soaks. Another set of experiments, we looked at SO2 additions. So we had a 0, a 20, and a 40. That's just total SO2 added. And again, we looked at these same cultures and their effectiveness against Hansenia spora once we added SO2 into the system. There we go. So I'll just walk you through this series of results. So here we have on my right, your left, the growth data for both Hansenia spora by itself as well as Hansenia spora in combination. So at 10 degrees C up here, the black square is Hansenia spora by itself growing. So at 10 degrees C, it grows pretty well. At the triangle bar here is the Mechnikova that we put in a mixed culture with Hansenia spora. It also grows well at 10 degrees C, even in mixed culture. And then here is where we had them both together. So you can see when they're both together, you have a lower growth of Hansenia spora, about a log and a half difference. So there was definitely some repression of growth even at 10 degrees C. We saw a similar thing, but not as pronounced at 13 degrees C. So even at the pretty warm temperature, we still had repression of Hansenia spora growth compared to if Mechnikova wasn't in the system. So it was fairly effective even at this warm temperature. And then if we look across to this table, we measured acetic acid at the end of the 6-day cold soak. And you can see at 10 degrees C, about 280 milligrams of acetic acid, almost double that amount at 13 degrees C as maybe we'd expect. But you can see once we added the Mechnikova in at both 10 degrees C, we got quite a dramatic drop from 280 to 100, and then at 13C from 435 to about 165. So you get the drop in growth, and then we also saw a decrease in acetic acid production. So that was encouraging. We repeated the experiments using a different yeast, the Torulaspora, and we saw slightly different results. So at 10 degrees C, again, if we look at the two squares here, that's the with and without and Torulaspora, you see a slight depression in the growth, but not as dramatic as we saw with the other culture. And at 13 degrees C, they overlapped. There was really no difference in growth. So less effective, at least from a growth repression. However, despite not seeing growth differences, we still saw a depression in acetic acid. So 300 to 171 or 440 to 235. So there was still this consistent drop, even though we would maybe not have predicted that from the growth. So there's still some suppression of acetic acid production, even if we didn't repress the growth like we saw with Mechnikova. So move on to SO2. We have all the graphs here for fructicola and SO2. The top is just the 0. And again, if you look at the two squares, you see that same repression of growth. Now, once we added SO2 into the system, so I'd say a low amount of SO2, 20 milligrams per liter, we saw a repression with Mechnikova still in the system. So even at 20 milligrams per liter, we still saw that decrease in growth. At 40, and actually the other thing to point out is Hansenia sporovivarum wasn't really affected by that small amount of SO2. You see the growth is still pretty high. But once we added Mechnikova in there as well, we got the depression. So very similar to what we saw at 0. Once we added 40 milligrams per liter SO2, then we started to see an effect of just SO2 by itself on Hansenia spora. And you see the square down here decline. It did come back near the end of that 6-day cold soak, but that amount of SO2, we were getting up into the higher amount where just SO2 itself obviously is having an effect on growth. As we'd seen before, in this case with SO2, once we added up the higher amount of SO2, we saw a depression in acetic acid. We didn't really see that with a low amount. But once we added fructicola into the system, we immediately saw a decrease in the acetic acid with or without SO2. Dalbruchii, as with temperature, seem to be maybe less effective as fructicola with SO2. Here, we don't see, again, much of a difference in the growth when we have SO2 in the system. At 40 milligrams per liter, again, that was enough by SO2 by itself to start having an effect. And so adding the other yeast on top of that seemed to help a little bit, but wasn't quite as effective as we saw with Mechnikova fructicola. So overall, with the model grape juice systems, we saw still pretty effective at warmer temperatures, particularly with the Mechnikova, and medium to higher SO2 amounts seem to also be effective in combination with the yeast as well. So we kind of took that knowledge and decided to focus in on SO2 when we did our actual cold soaks with Pinot Noir grapes. So we did 2 experiments, one with Mechnikova, and then we repeated it with Torulaspora. We had Pinot Noir grapes from 2020, and we did a 6-day cold soak at 10 degrees C, so slightly warmer than our previous work at 8. We really wanted to challenge the yeast in grapes. And we did 4 different SO2 levels. So we expanded on the SO2 and did a few more different levels at levels that may be a common addition up to a high addition for cold soaking. Same yeast as before and same Hansenia spora. We tracked populations, we measured acetic acid, and this time we also measured ethyl acetate. So Michael Chen's lab at OSU measured all the ethyl acetate for us, which we're really interested to see. The acetic acid the numbers weren't super high. We saw the trend, but the ethyl acetate, which we think was the more impactful spoilage compound we wanted to measure in this particular experiment. So at low to no SO2 additions, here's the growth of Hansenia sporovivarum. And so these are the colored, the full ones are when fructicola is in the system, and the open ones are when it's just Hansenia spora by itself. So if you're just looking at the effective SO2, you'd look at the open ones, and then the closed ones is the combination. At these low levels, low 0 and 25 total add, we didn't see much difference in growth, whether the SO2 was added or whether the non-Saccharomyces was added either. So the growth was pretty similar. There's one kind of outlier here, but really by the end of cold soak, they all kind of had reached pretty similar populations. When we upped the SO2 to 50 and 75 total add, so on the higher end, that's where you started to see really the effect of just SO2 by itself. So at 75, for example, we had our initial populations and then we never saw them again with our plating. The 75 knocked everything back out. But 50 was interesting in this case. 50 by itself, we still saw this low population. It didn't knock it out completely, but when we added the Fructicola as well as the 50, it knocked it back completely as well. So there was this added effect of SO2 and the culture. As I mentioned, the ethyl acetate one was really interesting to get that additional piece of information, and it proved pretty interesting from a results point of view too. If we have a look over here at the ethyl acetate column, you see with no SO2 and just Hansenia spora added, quite high ethyl acetate. So 116 milligrams per liter, that's well over the sensory threshold. It smelled very solventy, kind of, if you've ever smelled that coming off a cold soak or early ferment, it was pretty obvious. Once we added some SO2, we did see some decrease. And once we got up to the higher amounts is where, you really knocked that back considerably. But most interestingly to us was if we just added, the bar is a little out of whack there, but if we just added Fructicola, so no SO2, we knocked it back to about 10 milligrams per liter as well. So very similar to what we'd seen with the higher SO2 levels. So the Fructicola, even though we didn't see such a dramatic difference in growth, we still were having an impact on the production of the spoilage compound. And overall, there was this interaction between SO2 and Fructicola. As with the model grape juice system, the Torulaspora delbrueckii culture wasn't quite as effective. It still had an impact at 0 and 25, again, not really much difference between the growth of these cultures. Once we got up to the 50 and the 75 is where the SO2 had a stronger effect, with 75 being enough to knock everything out. And then the 50 was where we kind of saw this split. The ethyl acetate, not quite as high in the second round of experiments, but still quite high. I'm sorry, I can't quite see that, but that's 51 right there compared to 66. So Torulaspora by itself wasn't as effective at reducing the ethyl acetate production as the Fructicola was. So we saw some decrease, but it was more related to whether there was SO2 in the system as well. So it was less due to just the yeast. So overall, with the Pinot Noir cold soaks, as we'd seen with the model grape juice, the Fructicola culture was more effective, particularly in combination maybe with a low or medium SO2. So key findings, just as I kind of summarized, overall, both of those cultures, we saw reduced acetic acid production, but really the Fructicola culture was more effective at reducing growth, particularly over the temperature and then the SO2 concentrations we looked at. In the Pinot Noir cold soak, SO2 by itself will obviously eventually knock out Hansenia sporovivarum at the higher amounts and will reduce ethyl acetate. But in this case, we saw just the use of this culture was as effective as about a 50 milligram per liter add of SO2. And that combination of those two was also quite effective. So back to kind of some of our original questions around, could we use it as an additional tool with SO2, either reducing SO2 use? It seemed like particularly the Fructicola culture was still quite effective at reducing ethyl acetate in particular during these cold soaks. So, and our conclusion, a lot of these, they have some bioprotective activity, and in particular, the Fructicola culture we were using seems still effective even when there was a low SO2 add as well. I'd like to thank, firstly, the Northwest Center for Small Fruits Research for funding of this project. It's been helped fund the graduate student who did most of this work, along with a couple of undergraduates, and then Michael Chen for the ethyl acetate analysis, and then FRA, who's was in my lab at the time, Aubrey Dubois. And I'll take any questions.
So I hate to cut off the questions, but we're over time. And
Okay.
So I'm gonna,
Or I'll take
you have
questions afterwards.
Yes, that would be great. Yeah, thank you so much.
Okay,
So good afternoon, everybody. I am going to talk to you today about rose stem girdler, which is an emerging pest of caneberries, and this project that we did along with my colleagues. Click. It's not advancing. Should now? Okay. All right, so rose stem girdler, this is a buprestid beetle. It's a metallic wood boring beetle. The larvae feed on the cambium, and this causes a girdling effect. Emerald ash borer is probably a more commonly known agrolith species. With rose stem girdler, the hosts are rose family brambles, so we're talking about the genus of Rosa and Rubus. It's a pest of caneberries and ornamental roses. It's been long-standing in Europe. It's native of Europe. And the first reports were in the eastern US in 1878, and it was reported as well-established by 1913. And most of the information that we have on rose stem girdler and its control comes from the 1950s and the 1960s from two studies in Utah by Davis and Regavir. It was confirmed in Idaho by the 1970s and in Oregon by 1994, and then confirmed in Washington by 2014. From Oregon northward to Kane County, and then also in Spokane County in eastern Washington. Reports are just increasing in the greater Willamette Valley. As far as I know, it's still a current ORBC priority, and it's essentially naturalized at this point in Southwest Washington and Northwest Oregon. When we started surveying in 2018, a lot of indications were suggesting that the life cycle and the ecology was differing in the Northwest than what we were getting out of Utah, with later emergence and a longer activity period than Utah. Partly this might have to do with the fact that we just have really abundant wild hosts, Himalayan blackberry, evergreen blackberry, wild rose, thimbleberry, and then cultivated roses, think Portland, the rose city, right? And then the Pacific Northwest, caneberry and nursery industry, so a lot of hosts. Some of the main challenges for Northwest growers is determinants of risk are pretty unrefined or have been pretty unrefined. So do I spray at all? It's difficult to determine presence. It's really hard to scout for in real time. It's really easily missed, even when you've done it a lot. Signs of yield loss may be limited, variable, well hidden. And then growers can end up getting blindsided when you get a big infestation that comes in with significant damage to new plantings especially, but also just yield loss in general. And then IPM thresholds are not established at all for this. There's a really narrow effective insecticide application window that begins only after emergence, only after adults emerge from the canes, but before egg laying, which happens about 7 days later for at least that first generation that comes out. And emergence timing, which is totally key to this, is really difficult to determine. It doesn't adhere to a calendar date. It's biological. Of course it doesn't, right? And then once rose stem girdler is established, eradication's probably unlikely and probably unrealistic. So you have to know how to manage it once it gets there. So our first objective was to refine these risk determination aspects of RSG damage to caneberry plantings. For instance, right here with wild host, Himalayan blackberry next to a planting, how far can it move into a field in one season, for instance? But also to just try and establish, has it gone beyond the area that it was found in 2014, especially in the Northwest Washington, where most of our raspberries are produced. So for part of this, we surveyed 12 sites in Northwest Oregon and Southwest Washington, 9 grower sites, 2 experiment station sites. We put up, we collected damaged cane samples for objective 2, which I'll get to. We put up temperature and relative humidity data loggers at each site. And we put sticky traps with colors that have worked for other buprestid pests in the past to see if we could catch them that way. And we also scouted these weekly between April and August, looking for emergence specifically so that we had that data. We also continued to scout the 2 experiment station sites in about May through June 2022 for emergence again. The hosts that we found, of course, Himalayan blackberry, but also thimbleberry, which hadn't been confirmed before. And then evergreen blackberry and whiteberry raspberry had never been confirmed before either. The only one we didn't find it in is salmonberry. We haven't seen it in that yet, but we wouldn't be surprised if we did. We also just by chance scouted several retail nurseries thinking that plant material might be one way that this spreads, but we didn't find any row stem girdler. We did, however, find row stem girdler at a community plant sale. So that could be one way that it spreads. Some of the findings is that the traps were really not effective. No patterns, really weak indication, just no good method determined. We even did beating trays, and they didn't work well as well. The best thing to do is just look, keep looking, because it's just easy to miss. Determining the distance that these girdlers can move into a field from wild hosts just proved to be really difficult. We only had about 2 of 9 grower sites that were even marginal for determining the distance. It appeared to be about 300 feet, but the data just wasn't very satisfying in the end. We tried an alternative to do this. We took an experimental planting that we had at the Vancouver site, which is where I'm stationed, and tried to better establish this. So this is a 0.35 acre planting here. It was established for something entirely different. But it had 2 potential row stem girdler sources, one here and one here. This is a bunch of canes that we piled up right there, and this is just wild hosts that are infested right here. And the idea was to remove this closer one in the second year, so 2022, to see if it indeed was coming from here or if it was able to come that far in a single season. We sprayed weekly because this was the first year. We sprayed weekly during 2021 just to assure that we actually got establishment and they didn't decimate the planting. We also scouted this weekly between May and July. We actually didn't see row stem girdler in this planting, but damage was present nonetheless in late summer. Most of the damage was about 300 feet or less. So this hot spot in the middle of the field here, I can't see there it is. But some as far away as 415 feet, which is this little spot over here. In the 2022 season, the near source was removed. All the damaged canes from the previous year were also removed. So really the only source could be this or another one that was way far away, about 1,000 feet away. We did not spray that year. We didn't even scout it. No capacity. But row stem girdler damage that started to present in late summer was absolutely extensive. Like the whole field looks like this. We haven't even taken the data on it, but I know it's going to look like this. It's just going to be red. We also scouted 63 sites throughout Northwest Washington. And the first confirmation north of King County was in May 2022. So it is in Whatcom County. And then my colleague went on to find two other sites in Snohomish County, 8 more in Skagit County, and then 1 more also in Whatcom County a little later in September. Our second aspect, which highlights the importance of this, is to develop an enhanced IPM program with refined information on the emergence timing of row stem girdler and the development of a emergence prediction tool. And we did this through a controlled experiment where we collected all those canes from all these sites and then split them up into 4 incubator chambers with different temperature and humidity combinations. These are the phenology thresholds that Davis and Regevere came up with in 1963. And what we did with the incubators was mess around with these thresholds. They were outside, so all these canes were exposed to the same conditions up until 3/24/21, and then they got put in the incubators and they went under several different conditions, either optimal temperature or optimal humidity according to these thresholds. And I'll just highlight the suboptimals so you can see the contrast. Suboptimal humidity, suboptimal temperature. So this is what we saw as far as emergence goes. The suboptimal temperature incubators, we actually ended up turning up, I pushed the wrong button. We ended up turning up on the 21st of June because nothing had emerged from it. So in a sense, it worked, right? We did see emergence from both incubators with the optimal temperature. And then after we had turned this up for a while, we saw emergence, as you might expect, but much, much later. The thing that sort of is the take-home message, though, was that the humidity aspect just didn't delay emergence. It just didn't seem to have any effect at all, which was very different than what Davis and Regevere came up with. And so we wanted to build a model with these two, with the data that we collected from the outdoor scouting versus the indoor incubator conditions. And these warm colored bars that are going across like this are the indoor data sets. And then these ones that are kind of coming across are the ones from the outdoor sites. And what we're looking at is we want to find the point where these are converging, which is about right here. And this is just the same data set, just showing the variability a bit better, illustrating that a little bit better. And the temperature threshold that we ended up going with was 55 degrees Fahrenheit, 900 hours from January 1st, which is a safe dormant period for row stem girdler. And we were able to get this onto AgWeatherNet. It's now in beta mode, so it's still buggy at this point, but we expect to have it up and running more smoothly fairly soon. The thing with this is that compared to the recommendations that were originally in the PNW Pest Management Handbook from 2017, this model would have saved approximately 10 sprays per year in an average year. And then last year, which was the cool season, up to 13 sprays. So pretty significant. Our last objective was to assess the biological control potential of parasitoid wasps in reducing risk of girdler damage to caneberries and maybe its potential role in an IPM program. So we started finding as early as 2018 these ungirdled blackberries. So they had damage, but they weren't girdled. And then we wouldn't find a viable row stem girdler in it. And sometimes we would find these catches of eggs, or not eggs, larvae in them. So we started rearing them out. This is one of them compared to the size of a row stem girdler. This phenomena of finding canes that were damaged but not girdled was also reported back in 1940, just this one other instance as well. We were able to positively identify, it was much more complicated than I ever expected. And it's Baryscapus reglesi. It's in the tetrastinae. I can never say this when I'm up in front of people. This subfamily. And it includes known parasitoids of emerald ash borer in North America and row stem girdler in Europe. We reared out or caught probably about 60 of these wasps in total between 2018 and 2022. All the wasps were identified as the same species, which was first described in 1919. It's known to be a parasitoid of several North American native agarelus species, including red-necked cane borer, which is a native pest of caneberries in the eastern U.S. It was noted one other time as a parasitoid of Rostum girdler in New York in 1940, but never found on the West Coast to date. So this is the first find. And it's never been employed as a biocontrol, and no other existing information on levels of parasitism exist. We are going to try and collect that data, though, from wasp emergence holes on the canes that were in the incubation chambers. So a couple major hanging questions. We have, how can Rostum girdler presence be more easily detected? Because, you know, this is a key component of an IPM program. Is it there at all, right? Also, what level of control is this WASP capable of? And can it be actively employed as a biocontrol agent within an IPM program? So with that, I'll take any questions. My phone went off, so I don't know how much time
We have about 1 minute for questions.
Okay.
Was Benton County lit up on your map? Oh, sorry.
I don't think so, no.
Well, they're in Benton County.
Good to know. Good to know.
Yep. And in Southwest Portland. I had one on my window screen
Yeah.
this past fall. And at first I freaked out. I thought it was an emerald ash borer.
Yep.
Right. And I started, because I'm familiar with that from Michigan.
Interestingly, they did not include that in the lookalikes.
Interesting.
But I let them know.
Yeah. But then I looked at it closer and it's like, that's the
EAD.
adult.
Yep.
Yeah.
Yeah, they're all over Portland.
And we found them in Benton County on black raspberries.
Okay, good to know.
Wait a second. Any pheromone? You working as a pheromone? Application is pheromone?
Oh, a pheromone trap? That would be wonderful. No, we didn't do anything with that in this. It was just color. It was just color. That's all we did for this. We don't have a pheromone for it, so that would be for another project. All right.
Idea and the concepts. Basically, the idea is to apply double-strand RNA molecules. I think now everybody is kind of aware of when we talk about RNAs with the COVID and the pandemic. So the idea is actually to trigger through the application of this double-strand RNA an immune response to the plants. And the plant does not have any antibody systems, but they do have what we call RNAi interference. Okay, so the idea will be to apply that and to target either some pest or some aspect of the plant, some endogenous genes of the plant, and of course, in the hopes that there is no any over insects or even human being targeted by the applications. So the idea is to provide the double-strand RNA and to basically have formulations with some nanoparticles to protect and facilitate the uptake of this double-strand RNA into the plants. Next. Oh yeah, next. So you are going to hear a lot of next. Huh? So the concept itself is, as I said, based on well-conserved mechanisms named RNAi interference, which basically is triggered by the presence of double-strand RNA. This is a co-evolutionary. Go back. Here, let's here. So we do have the mechanisms is actually triggered by the presence of these molecules that can be actually integrated into different path mechanisms, leading to either the methylations of the target DNA of the pathogens or the endogenous plants, or the cleavage, or the repression at the protein level. So with the cleavage, you have a decrease in intracellular abundance of the targets. With the translation inhibitions, then you don't have actually the protein that is synthesized, and you have an increase. Basically, this is an increase of the transcripts. Next. So that is the biological aspect. Now, the idea is to provide the double-strand RNA with a nanoparticle that will facilitate the delivery of this kind of cargo molecule to the plants, protect those molecules under harsh environments like a leaf surface, and facilitate the uptake. Next. Okay, so we decided to go with some sort of very cautious way to prove the concepts, and we focused on actually powder mildew. Why? Because there are some foundational knowledge about powder mildew and what are the genes that could be used to confer some resistance. And because the idea would be to actually silence the genes, so knock down the genes, we went through a very specific class of, yes, go ahead, of genes that are MLO genes. They are what we call X-ray susceptibility genes. So that means when you knock out, they tend to confer resistance. So how do they work? Basically, MLO tends to negatively regulate another class of proteins that are essential in the formation of the papilla when the powder mildew is actually invading the plant cells. And there are evidence the transgenic through some DNA recombinant technologies that transgenic lines of some of these MLO tend to actually confer resistance relative to high resistance. All right. So next, as a matter of fact, next, there is a group of, this is the phylogenetic trees of the whole MLOs that exist in the genome of grapevines. And you have a group here, the CLED5, that contain all these candidates. And those have been validated. And there are 2 others that were part actually of that clade. So they are pretty closed. Next. So that was actually from the plant side. Now we want also to look at the fungus sides, and then we went with very basic life cycle related genes that are essential to the integrity of the pathogens. Okay, and this one is basically that gene is associated with the ergosterol synthesis, which is a critical component of the cell wall. Next. The other aspect we decided to target is basically also fungus. Fungi tend to actually have their own RNAi mechanisms and then tend to basically deliver some small pieces of small interfering RNAs to the plant to basically repress the RNAi mechanisms of the plant. Okay, so this is kind of the home races here. So we have genes from the plants and genes from the pathogens. So based on that, we decided to develop those projects next. And we delineated 3 main projects. Identify the double-strand RNAs that basically from the plants or from the fungal target genes capable of silencing their own expressions. Then evaluate the plant's uptake and processing of double-strand RNA to assess the systemic movement of those molecules within the plants. And we examine the use of one specific nanoparticle, nanoclay, as a nanomaterial to efficiently deliver the double-strand RNA to grapevine leaves. Next. Okay, so from objective 1, the idea was first to identify the target genetic regions for the production of RNA. And this is, we went through actually some bioinformatics, not bioinformatics, but just protein alignments and also RNA alignment to identify some potential regions that could be used to generate those double-strand RNA. You can see there are some overlap between all of them, but when we look at the sequence, basically there are none 21 to 26 nucleotides that were common across these overlap regions. So we think that it was good enough for us to consider that each of those double-strand RNA could have a gene-specific response on their targets. Next. We went through some tools that are available online to basically predict the number of potential candidates that could be existing through this double-strand stretch, and we used that tools that was recently developed here to have some sort of confidence about, yes, what we see in using those regions seems to indicate that they will produce some small RNA that could be part of an RNAi mechanisms. Next. So now we went through actually the approach. Logistically, we need actually a systems where we could propagate easily the plants and in a controlled environment, just for proving the concept of the silencing. And in that case, we opted for a root soaking experiment from in vitro plants. So this is some plants that we are using in the lab. And basically, just by transpirations, basically, we have the double-strand RNA mixture in the solution, liquid solutions. They will be absorbed, and then we could potentially look at the impact on those endogenous plant genes of MLO. Okay. No, good. Go back, sorry. So this is the experimental design. So we basically collected leaf and root tissue after the incubations with the mixtures, and we collected at different times. We have mock plants with no double-strand RNA and with double-strand RNA, and we did some real-time PCR to look at whether or not there was some silencing. Next. Okay, so first of all, for MLO3 and 4 that are part actually the clade 5, we could not actually see any change in transcript abundance in the root and the leaf tissues. Now for 6, 13, and 17, we could see in both tissues actually some changes in the transcript abundance. So look at actually the day 0 and then look at actually the evolutions of the transcript abundance for these different genes. And what we, next please. What we observe is actually an increase in some instances to pretty dramatic fold change compared to the day 0. We were talking in some cases at day 3, for example, in the roots here, 10-fold changes more in the roots due to the treatment. So in some cases, we have actually decreased. So remember at the beginning, I was mentioning something about RNAi can lead to either degradations of the transcript or a repression of the protein synthesis. They could be basically leading to an increase of the transcripts because the system is actually stalled by the presence of the machinery. So increase. So basically, next, what we have is seems to have actually a response for MLO6, 13, and 17 using those stretches. In some cases, we have RNA cleavage for MLO13. For other, we have actually translation inhibitions. Needs to be actually supported by other evidence, but it looks like it is the case. Next. So the second objective was to look at whether or not these double-strand RNA in normal conditions could actually be could be absorbed and moved from cell to cell after the uptake. So, and this is, we did with actually a fluorolabel double-strand RNA with the Cy3 with treated roots. So here you have to trust me, but basically in this case here, there's the Cy3 fluorescence. They tend to actually accumulate on the root hairs of the roots, of the secondary roots, but also in the vascular bundles, which means that basically they are getting into the roots, and then after they cross and go from cell to cell up to actually the vascular bundles, which might suggest that they will get upward toward actually the upper part of the plant. Next. Now the objective 3, and here this is where we went through a lot of hurdle. We are not chemists, but we thought actually the simplicity of the mechanisms was good enough for actually getting something, and we have been challenged by actually this objective 3. So the idea here is actually to go through actually some sort of quality control steps. Okay, so first the synthesis of those nanoclay, then the quality of those nanoclay, and then checking whether or not they are able to load, whether or not the LDH can be loaded with the double-strand RNA for the protection effect. And then after testing the capacity of the formulation to be uptaken by the leaves. Okay, so you see that this is not very super chemistry. I mean, this is not super complicated. It doesn't change the fact that we went through a lot of problems. Next. We basically, the idea was to target a very specific range between 15 to 100 nanometer with a positive charge. And we have never been able to actually reproduce other papers. Those 2 papers are the 2 major papers, and we have never been able actually to do that. So basically, with so much backgrounds in chemistry, we have actually to come up with something, and we try several several systems, but most of the times we have now the particle size was actually too big. So there was something where we communicate with a lot of chemists and engineering people to actually better understand what happens. And finally, I think we are in the edge. We start to see actually the light at the end of the tunnels. We came up with actually a pretty straightforward protocol that lead us to something that is quite close actually to the range we are looking at, which is 20 nanometers to 500 nanometers in terms of the size, and the charge is actually still positive. So looks like that we are quite close to where we were shooting for. Next. Now, we were quite happy by that, but the thing is, here is what is happening. Here is actually the LDH from those papers, tend to actually have a very uniform size and quite dispersed, and this is basically what we have so far. So basically, polydispersed, different size, and aggregated. The aggregations, I don't have time to actually go through it, but we were able actually to find a way by sonication to disaggregate those particles. But it looks like that we still have different size in the particles, so that could have an impact on the loading. Next. All right, so the conclusions. Again, this idea here, I was not actually shooting for actually showing spray. The idea is actually to develop the phase one of the project before going into a field aspect. So the root uptake seems to be actually very nice, straightforward systems to evaluate the silencing. We have been able to induce the silencing for 6, 13, and 17. We failed with actually MLO9. We had some issue with the amplification of the transcripts. So this tend to, we were using actually not most of the alignments were done with actually the Pinot Noir genome, reference genomes, and we were using a different model here, the Macrovine, which is perfect for actually lab environments. Maybe some slight variants in the regions that we use for the real-time PCR could actually explain some of the discrepancy. And we need to revisit actually for 3 and 4 the regions that we have been using so far because we could not actually get any silencing. The synthesis of the LDH was, as I said, extremely challenging, but I think we are on the right path now. Next. And we are actually looking at another, it seems to be actually quite tedious as a work. No, go back. Sorry. We are envisioning and we are working on a new nanocarrier, which is actually the carbon nanodots, which is much simpler actually in producing, very inexpensive and with right away the proper size. That actually, this is something that we are quite excited to use in a very near future. Next. Okay, the remaining questions because of this different thing that was lagging. We are working on this aspect because silencing is one thing, but now we want to see actually whether or not the silencing actually is triggering the resistance, at least for the MLO genes, right? So we are going to go back to the root socking systems and checking whether or not after incubation with those dRNA, can we challenge the leaves of those plants with actually conidia? And I think we were able to come up with actually a good protocol for inoculating those in vitro plants. So that is good. Next. And now jumping on the fungal genes. We'll try to see actually if the ERISF indicator can absorb those small RNAs. So this is something that we are working now. Next. And finally, go to actually more mature plants and do the spraying the DNA, RNA, LDH mixtures or double-strand RNA, LDH, not LDH, but carbon dots. That's it. Next. I don't remember next. No, there is no next. Okay, thank you.
There's 1 minute for questions.
Sorry.
Laurent, did you use a single cultivar and was it susceptible, or you use multiple?
I use The cultivar for the susceptibility, you mean?
Yes.
For the next, this is that's a good question. I didn't look at this aspect, but the progeny of this is based on actually a Pinot Noir, which is, I think, susceptible to powdery mildew. So it should be not a problem, actually.
Okay.
Yeah, it's not a resistant for sure.
Okay.
Yeah. They grow very well. The powdery mildew is actually thriving. Yes.
Does that nanoparticles work out?
We right now the loading with the LDH is underway. The loading with the carbon dots is actually working. So basically, we have clearly actually a way to go with the carbon dots in terms of protections, and we are in the middle of testing now the formulation to the leaf. Now everything here is actually in the lab environmental conditions. We have not yet jump on fields, and we still have to look at actually in the greenhouse with mature leaves, which contain a lot of trichomes that could actually interfere in the uptake.
Okay.
Thank you.
Okay, next up is Sarah Loader, and I will let.
Okay, great. Thank you. I'm here on behalf of Kelly Vining, who's the PI on this project. And the student who worked on it is Christina Mull. She did her master's degree in this project and now graduated and went on. The project was initiated by Chad Finn, who made the crosses for the 3 populations we worked on. And our expert is Michael Dossett in aphid resistance in black raspberry. So it was a collaboration between all of us. So I'll be talking about identification of disease resistance candidate genes for the North American large raspberry aphid, Amphorophora agathonica. It's going to take me a while to spell that. Okay, so the reason we wanted to do this project is to reduce the rate of black raspberry necrosis virus spread in black raspberry fields that affect the lifespan of the crop. And so our objectives were to use messenger RNA sequencing to find gene expression differences in plants with and without aphid inoculation, between resistant and susceptible plants, and plants harboring each of the 3 different genetic sources of resistance that were previously found in our previous research. And we wanted to develop and test molecular markers targeting these genes with roles in resistance. So the populations that were developed were between, so these were the populations initially that were used to map the resistance locus. And so we used an offspring of each of these populations here where the star is that are resistant from the source. So in this case, the source was from Ontario, in this case was from Maine, and then from Michigan. Those were the 3 sources of resistance to aphid. And we had the same susceptible parent ORS 4580-2. So the 3 crosses are here, and you can see that they segregated 1-to-1. Basically, half of the offsprings were resistant and the other half were susceptible, confirming that it's a single gene in each case that controls the resistance. So the questions were, how does gene expression change when black raspberry plants are attacked by this aphid? And the second question was, how do gene expression patterns differ between aphid-resistant plants and aphid-susceptible plants? So what we did after we collected the RNA is we did sequencing, and we used 2 different platforms. One was Illumina for short read, so it sequences short segments, and that's used to study the levels of gene expression because we get high coverage, high depth of these small short sequences of DNA or RNA in this case. And then the long read sequences, it's used to find the full length transcripts because they're quite large of the expressed genes and the potential variants for those long genes. Okay, going to the results, we saw a general trend of decrease in gene expression after the aphid inoculation. And then there were sets of genes that were specifically expressed in each of the different sources of resistance. So in this case, we have 141 genes that were only expressed in the main source, 24 that were only expressed in the Michigan source, and 77 in the Ontario source. There were a few that were in common, but not a single one was in common between the 3 different sources. And you can see here candidate genes that are similar to, that have a function either in stress response or disease resistance in plant defense in the 3 different populations. And those would be targets for developing DNA tests for resistance that can detect resistance. So the genes with the greatest increase in expression in resistant black raspberry plants, they seem to function in floral timing and in development, as you see here in this table. So this is, and you have all the different sources, Ontario, Michigan, and Maine. So you see this ubiquitin ligase gene, a zinc finger constance-like gene that's a transcription factor, an early flowering gene. So basically they function either as transcription factors or in floral timing and development. So we would like to develop and test DNA markers targeting differentially expressed genes and segregating black raspberry populations next, and we didn't get to do that. And I'd like to thank the Northwest Center for supporting this project, and I'll take questions. Yes. How do I describe them?
Yes. How you know this is aphid resistance or non-resistance? How we know that?
Oh, you mean how do we know that the plants are resistant and the plants are susceptible? So what Christina did is you put 3 aphids on the young seedling, and then you observe if they stay or they go, or they don't like it. And then you do it multiple times. So if you don't find any, then you come back and then you put 3 more and you observe them. And so it's very, very clear. Ones that are resistant, the aphids don't stay on them. The ones that are susceptible, you see a lot of aphids on that plant. And Jana Lee can tell you. They're very, very clear responses. Sorry? Yeah, they don't stay on them. And actually, thank you, Jana Lee, for reading the aphids. Jana Lee she collects the aphids for us and reads them so that we can use them for that. It's a clear, and Michael developed that assay.
Once the aphid attack to, crop, the gene is turned on. That possibility, or the crop, resistant crop, already this gene is already expressed.
Right. So, we did also look at the difference prior to inoculation between resistant and susceptible, and we didn't see anything. We need someone who has very good bioinformatics skills to really delve deeper into the data, because there is a lot of data. And I think Michael Hardigan may be convinced to do that. Yes, because there is a lot more work to be done, unfortunately, or fortunately, I don't know. Any other questions from anyone? Okay. Well, thank you.
So we do have a little bit of extra time. So if James wants to answer any questions, yeah. So feel free to come up. But if not, we will be ending this about 8 minutes early, which is okay. Okay. Well, thank you everyone for attending this session this afternoon, and really enjoyed hearing about everyone's research. And with that, I will see you all at dinner.
On mute. Can you hear us now, Sarah?
Oh, I can hear now.
Oh, perfect. There we go.
Awesome.
Cool. Whenever you're ready, you're good to go.
Perfect. I'll go ahead and share the screen. Can you all see that?
Sarah, you should have a view of the screen now. There we go.
All right. Well, excellent. Thank you all very much for listening. I'm excited to tell you a little bit about this project. So first, cooperation, a common problem. And this can describe many different kinds of scenarios, scales of conversation between two growers that could be talking about a particular pest issue that they're both experiencing. Pest quarantine for either county or country. And pests and diseases are often such common problems that sometimes need some level of cooperation to properly addressed. And these are because pests and diseases do not respect property lines and can potentially spread for long distances. So how others in a region manage those pests can really affect an individual's pest problems. And while cooperation can scale based on the complexity of the actions required to manage a collective problem, it can also scale based on the number of people involved. While two people sharing information on pests and diseases can absolutely count, it can also scale to larger information sharing systems such as spore or monitoring, insect monitoring networks, or technical groups. But information sharing is just the simplest form of cooperation, which is also the backbone and first step to any further and sometimes more complicated actions. But information sharing is also very common in agriculture overall, with viticulture being no different for this. However, while many growers engage in some form of cooperation, there's really not very much information on how this engagement occurs or how to potentially increase engagement in any sort of cooperative management strategy. So the objectives for this project were asking the questions: what do growers think about cooperative management, and how do those opinions rank in their importance for all the rest of the things that they have going on? And how does that information pass to and among grape growers? Therefore, we sought to investigate this through a series of surveys. This started just by talking to growers and asking several open-ended questions about what matters to them most about pest and disease management. I don't know if I heard a sound. I'm not sure if that's anything. But we took those responses and grouped them into common themes and summarized each of those unique opinions and points into a set of statements. For the second phase, we came back with a separate set of growers that we wanted to learn about how they were prioritizing all of these statements and opinions And then those priority rankings focused on either information resources or cooperation in management. So the first set of surveys on our opening questions was asking about grower thoughts on cooperation and pest and disease management. And many of these answers centered around either crop health concerns, such as vine and soil health, economic concerns, such as prospects on return on investments or labor availability, as well as more social and behavioral concerns, such as the differences within the group and administration around cooperation. So for this survey, we generated 36 statements that touched on the issues listed here. And so for that second group of participants, that second phase of the survey, we took those 36 statements and sorted them into an arrangement like this shown here. There's a lot of text on this slide, and we don't get to go into all of these details, but just each statement would go into a box where they were sorted based on whether you agreed with or disagreed with the statements and how much you agreed or disagreed with the statements. So here we assume that participants would agree and disagree with roughly half, which may not necessarily be true, but it did allow us to look at some of the more priorities and how people prioritize that information more readily. And the responses for those surveys were collected and using a principal component analysis. We were able to identify some of the major trends and perspectives that emerged from those responses. And for the cooperative management group, we were able to find there were four major perspectives, four major archetypes, as what we call them, that really explained much more of the variance that than any of the that was found based on the demographic information that was also collected. That looked at location, acreage, experience, management philosophy, that sort of thing. So what we're seeing is just, despite the group that was surveyed, we really are seeing much more variation within these populations and than we are necessarily associated with the demographic information, that there's a lot of variation within these demographics. And while I don't have too much time to go into detail here again, overall, we also saw that participants still were generally positive about cooperation statements. For example, the statement, "Cooperation is important because pests and diseases do not respect property lines," was mostly agreed with, while the statement, "I do not feel regionally coordinated pest management efforts are worth the time and effort," was the statement that was the most disagreed with overall. However, despite feeling positively about cooperation, participants really were still more concerned about their own individual crop health and economic concerns, which this is not necessarily a revelation necessarily, but it really does emphasize that if there is an intended cooperative management action in a region, the potential participants really need to perceive that they are perceiving a personal benefit from that action in order for them to participate. Then we're going to transition to looking at some of those information resources. So when we're talking about how do you look for information on pests and diseases, how do you get that information? We got, again, this qualitative survey, we were able to see 23 different items here, which were kind of indicated by the smaller text on this image. But we really saw some more overall trends that were discussed, either like in-field information versus out-of-field or regional, that looks at information that's either directly from your vineyard or from outside of that in the surrounding region. Then there's other general scaling of information resources, looking at either as small as up to your neighbors, ranging to a much larger scale of international resources and everything in between, as well as active and passive communication. And that's whether the learner might be expected to participate more or less in that learning process, respectively. So like a conversation versus listening to a talk. And again, participants sorted these statements with one statement per box based on their level of importance for this one. So this example arrangement, the most important resources are over at the top at 8, where the least important are over at 0. And once again, we force them to rank into a normal distribution where fewer options are at the most extremes with more in the middle. And this again allows us just to look at more of how that information was prioritized. And again, we're going to just very briefly get into these results. But I just wanted to mention our most highly ranked resources here. The professional network, in-field data, field foremen and supervisors, and direct contact with extension were the top overall sources. I mean, while we did see some variation, really we saw that social and experiential learning was really emphasized as the most important and most useful to growers as they were learning about this information. And so there's a third survey looking at viticulture communication networks. Essentially for this survey, we asked, with whom do you talk about pest and disease management? And participants could provide the names of either growers or outreach professionals, though those are not mutually exclusive titles. And we primarily focused on the Willamette Valley of Oregon, though we had some participation in Southern Oregon, the Columbia Valley of Washington, and Napa Valley, California. But participation was not restricted to these areas. But that's kind of the general areas we focused on. And again, we had about 65 participants for this particular survey, which does not even get close to full saturation for these regions for sure. But it does allow us to still begin to look at some of the communication network and social networking structures that are involved here. And specifically, we looked for structures like cooperative triads and structures that were associated with information diffusion or the presence of popular actors. And so for these structures, we had two different methods to examine them. Either egocentric random graph models, ERGMs, which used an MCMC, maximum likelihood estimation, for look at all three of those parameters. And then random graph comparisons, which just took a random graph of the same size and compared whether that process was going to be geared from social networking processes involved. So cooperation triads, these are a closed network structure that are involved with three actors just linked together. So all three of them are communicating with each other. And these are associated with cooperation because they help promote trust and reciprocity in a community. And here we have a positive GWESP parameter, which indicates that we do have a tendency towards triadic closure. However, it is less than we saw in our simulated random networks, indicating that we have a mixed level of support for these structures. And then information diffusion is intended to describe how long it takes for information to pass through a network. So like the game of telephone. So here we have the least efficient and the most efficient pathways for this. So either it has to go through every one individual, which will take much longer, or it can go through a central individual. So the longest path between any two people is 2. And our HMPL harmonic mean path length just describes the mean length between these two. And what we saw here is that we have a much shorter than random HMPL, indicating that we do have much more efficient transfer information in these networks. Then we have spatial homophily, which is just examining whether actors are much more likely to connect with others within their own region, which we might expect this due to both physical distance as well as more similar regional problems and conditions. And we do see this because we have our positive parameters here, as well as the clustering of the colors, which are indicating the state on our sociogram. So the green is our Oregon participants, blue is our Washington, and yellow is our California. But one thing I do also want to point out is that despite the fact that we do have a lot of spatial homophily, we do have a lot of regional clustering, we do still see connections between all of these different regions. So we are getting information passing through all of these separate regions as well. And then we have the principles around central figures, which are very much related to information diffusion. But this is looking at specifically whether information is passing through the same set of popular actors, which can help narrow down the number of individuals that an outreach professional has to target in order to diffuse a certain piece of information. And we have negative link parameter here and degree centralization values that are significantly larger than our random networks, which are all indicating that we do have fewer individuals that information is passing through. But we can also look at those popular actors in a slightly different way. For the networks previously discussed, they have made the assumption that it's undirected, because we assume that if you and I are having a conversation about pests and diseases in the vineyard, that information is passing both ways. However, if we use a directed network, we can look at the individuals that are more influential in a network. So while the number of connections overall, which is the degree centrality, is shown here by the size of the icon, the more green that icon is the more that particular actor was specifically identified by others. So while these networks do not describe our entire viticulture community in these regions, it still illustrates how few people can be used to reach many others in that network. So conclusions we can make here is that information sharing is extremely important to the viticulture community in the Western US. And I expect the same is true elsewhere. And we found evidence of structures that support information diffusion, central figures, and cooperation. However, because we did see mixed evidence surrounding the cooperation structures, this may indicate that if a more intensive collective action effort is needed in a community, there likely needs to be more support structures in place to make sure that participants feel more comfortable and therefore more willing to engage in that cooperative action. So with that, I would like to thank the co-authors on this project, Michelle Moyer, Monica Cooper, Jay Scheidt, and Walt Mahaffey, and as well as the entire foliar pathology lab, which has been great, and the funding that came from the Northwest Center for Small Fruits Research. And with that, I would love to take any questions.
So based on one of your last figures, it looked like there were a few outreach professionals that were highly influential. Do you know who those individuals are?
So I do.
Can
Technically,
you
this
share
is-
that, or is that?
I can't necessarily share that because this is an anonymous or a privacy protected survey.
Okay,
But
understandable. But it's good to see that the outreach professionals are very effective.
yeah, for sure. For sure.
That was a very interesting talk, Sarah. Thank you.
Thank you.
I was wondering how this information is going to be used. Are there some areas of communication that could be improved? And if so, how?
Absolutely. So I didn't get to go into a lot of detail about some of the archetypes that we also learned from this survey, just because it takes a lot to get into that, and we didn't have time for that. But through that, it kind of gives you a sense for the different information resources that certain people like better. So while we did see a trend where people really found their networks very important, there were differences, clustering of differences on the particular resources that they use. So some people really liked using just their state extension publications. It was one of the first places that they would go to look at, and did not necessarily look at AVA information, where others tended to go more central towards that. And that sort of thing. So you can kind of get a little bit better sense of maybe how, at least as an outreach professional, you might focus your information production efforts so that you can at least get to the resources that people find more effective. It can also be used as people are, it's trying to help people who are maybe in the outreach game, is kind of where this research is focused, to help people deliver information more effectively to growers and help growers more effectively manage their products. So if you have a collective action effort, it's hard to do that without a better sense of your audience. And so this kind of helps provide some of that context as you're going to engage in some of these activities, if that makes sense.
Sarah, thank you so much. We're going to have to move on to the next presentation. That was wonderful. Thank you.
All right, thank you very much.
Yep. So next up is Justin O'Day. Is that? Okay, fantastic. And yeah, excited to hear your work.